Let's start our next chapter, chapter number 20, Biotechnology and Genetic Modifications. Here our key learning objectives are understanding microorganisms and enzymes applications, exploring genetic modification techniques, analyzing advantages and disadvantages of genetic modification in crop plants. Let's talk about historical context of genetic manipulation, early agricultural practices, selective breeding. Humans altered organisms' genetics for thousands of years. Began growing wheat and cereals crops over 11,000 years ago. Selected best seeds for cultivation. Improved livestock through strategic breeding. Scientific breakthrough occurs in 1953 milestone when Watson and Crick discovered DNA structure. Scientists learned to move DNA between cells, alter cellular DNA, manipulate genetic material purposefully. Now let's talk about genetic modification techniques. Fundamental concept and the definition here is changing genetic material in cells or entire organisms, called as genetic modification technique. Methods include removing genes, altering existing genes, adding individual genes. Applications include human protein production, insulin production examples such as we have human insulin genes inserted into bacteria. Bacteria produce human insulin. Advantages: larger production quantities, consistent quality, reduce reliance on animal derived insulin. Let's talk about crop plant modification. Types of genetic modifications. First is herbicide resistance. Example, soya plants resistant to glyphosate, allows selective weed elimination. Next is pest resistance. Example, cotton plants with Bt substance, reduces pesticide usage, protects crops from destructive insects. Nutritional enhancement. Let's talk about golden rice case study, addresses vitamin A deficiency. Genes from maize and bacteria inserted produces carotene, converts to vitamin A. Potential to prevent blindness and reduce mortality. Some ethical considerations, controversies, public suspicion about genetic modification, concerns about potential unknown risks, debate between scientific benefits and potential dangers. Regulatory approach, gradual acceptance by governments, safety registration process. Countries like Australia, Canada, New Zealand, US are having leading regulators framework. Fermentation process for biotechnology. Let's talk about fermenter components first. So the components include sterilization. It eliminates contaminating microorganism. Fermenter components. Second component is nutrient supply. Provide energy and growth requirements. Third is temperature control. Maintain optimal enzyme functioning. Number fourth is pH monitoring. Ensure ideal microbial growth conditions. Now let's talk about commercial products. It includes insulin, penicillin, and mycoprotein. Genetic modification. Overview of genetic modification. Definition first. Process of changing an organism's genetic material by removing changing or inserting individual genes. Widely used in biotechnology and medical research, involves manipulating DNA to create desired characteristics or produce specific proteins. Now let's talk about types of genetic modification. Bacterial genetic modification. Key applications: producing human proteins for medical use. First successful example is insulin production for diabetes treatment. Proteins produced by GM bacteria. Blood clotting proteins for hemophilia treatment. Human growth hormone. Interferon for cancer treatment. Now, crop genetic modification. Primary modification types: herbicide resistance, pest resistance, nutritional improvement. Let's talk about genetic modification process step by step. Gene identification. Locate specific DNA segment coding desired protein. Use restriction enzymes to cut DNA. Then plasmid preparation. Remove small circular DNA from bacteria. Cut plasmids using same restriction enzyme. Creates complementary sticky ends. Then DNA joining. Third step. Mix human DNA fragment with the cut plasmid. Use DNA ligase to connect fragments. Create recombinant plasmids. Number 4, bacterial transformation. Insert recombinant plasmids into bacteria. Bacteria reproduce in controlled fermenter. Express human protein. Let's talk about advantages and controversies of GM crops. Advantages: increased crop resistance, reduced pesticide use, improved nutritional qualities, potential solution to global food challenges. So these are all the advantages of genetically modified GM crops. Okay. Now, concerns and arguments against GM crops are: potential environmental impact, high seed cost for farmers, possible gene transfer to wild plants, and there are some ethical considerations as well. Key enzymes in genetic modification. So we have number 1, restriction enzymes. The function is cut DNA at a specific point. Its significance is it creates sticky ends for gene insertion. Next is DNA ligase. Function is join DNA fragments, and significance is it enables creation of recombinant DNA. Let's talk about some ethical and scientific consideration. There are ongoing research and debate on this topic. We need to carefully evaluate the potential risk. Balancing technological advancement is crucial with environmental and health concerns. Okay, now notable examples we have golden rice, genetically modified to increase vitamin A content. BT maize, pest resistant crop using naturally occurring bacterial protein. Herbicide resistant crops reduce agriculture chemical usage. Further future perspectives includes continued technological refinement, potential solutions to global nutritional challenges, increasing precision in genetics modification techniques. Biotechnology and microorganism. Let's talk about biofuels and yeast production. Here first we will speak about ethanol production process. The key concept here is yeast can break down sugars through anaerobic respiration to produce ethanol. Learn it. Primary crops are maize and sugarcane. Crop processing methods now. For maize, treated with amylase enzyme to break down starch to glucose, and sugarcane is naturally contains sucrose, requires no enzyme treatment. Biofuel advantages and challenges. Pros are sustainable resource, reduces atmospheric carbon dioxide, renewable energy source. Cons are lower energy density compared to fossil fuels, competes with food crop production, potential ecosystem disruption. Let's talk about bread making process. Ingredients and biochemistry. Key components include wheat, flour, water, yeast, gluten, protein. Fermentation mechanism include amylase enzymes break down starch. Yeast performs anaerobic respiration. Carbon dioxide production causes dough rising. Gluten provides structural elasticity. Let's talk about enzymes application in biotechnology. Pectinase in fruit juice production. What it does? Break down pectin in fruit cells, facilitates juice extractions, improves juice yield and processing efficiency. Let's talk about microorganism advantages in biotechnology. Key benefits are rapid production, controlled growth or conditions, genetic maneuverability, efficient protein enzyme production. Genetic modifications overview techniques: gene insertion removal, plasmid manipulation, restriction enzyme usage, recombinant DNA technology. Applications include human protein production, crop resistant enhancement, nutritional quality improvement. Let's talk about ethical and environmental implications, balanced approach to biotechnology, consideration of long term ecological impacts, sustainable development strategies. Let's talk about experimental skills and biotechnology. Here we are going to discuss about biological washing powder experiment design. Now, experimental planning methodology, you have key experimental considerations. Select a clear hypothesis, identify independent dependent variables, standardize experimental conditions, develop comprehensive safety protocols. Potential hypothesis to investigate egg strains removal efficiency, optimal temperature performance, stain removal effectiveness on cotton cloth. Experimental design checklist includes independent variable. You can use it for determining variation range, select appropriate concentration levels, conduct preliminary testing. Dependent variable, define measurement criteria, establish precise measurement technique. Control variables, maintain consistent experimental conditions, minimize external interference. Let's talk about safety consideration, precaution, use protective equipment, handle biological washing powders very carefully, follow laboratory safety protocols. Okay, now let's talk about enzyme functionality in washing powders. Enzymes characteristics. Biological washing powders contain specialized enzymes. These enzymes enhance stain removal capabilities. Early biological washing powders functioned optimally in warm temperatures. Enzyme origin. Modern enzymes often source from bacteria. Bacteria naturally adapted to hot spring environment. Natural selection drives enzymes development. Next is lactase, enzyme for lactose breakdown. So, what is the lactase functionality? It breaks down lactose into glucose and galactose, essential for infant milk digestion. Genetic variation in lactase production. Lactose tolerance. Genetic differences include European descent, lifelong lactase production, Asian descent, limited lactase production, potential digestive complications. Let's talk about lactose-free milk production. Enzymes treatment enables lactose-free dairy products, allows consumption for lactose intolerant individuals, alternative sugar production for confectionery. Let's talk about fermenter microorganism cultivation, fermenter components and their function. Stainless steel container, primary function, primary growth environment. Nutrient inlet, provides essential growth materials. Temperature probe, monitors optimal growth conditions. pH meter, regulates environmental acidity. Paddles ensure content mixing. Now let's talk about cultivation process. It includes sterilization. Steam cleaning eliminates contamination, ensures pure microbial culture. Nutrient provision, it includes water addition, glucose for energy, nitrogen sources for protein synthesis. Next is aeration, oxygen supply for aerobic respiration, waste gas management. Let's talk about temperature and pH management, precise environmental control, enzyme optimization, continuous monitoring and adjustment. Let's talk about harvesting, product collection after sufficient growth, extraction of desired microbial products. With this, we have finished all of our 20 chapters of biology IGCSE, and now we are going to start with past papers questions. Soon we will see you in our past paper questions and their solution. Recommended is to view all of the slides before working on the past papers.